This cover slide actually tells us all that I'm going to talk about. It's kind of, I can take an hour to talk about just this cover slide because it kind of summarizes the central dogma itself. Here, let me talk about it and I will move on with my other slides. You see that the core of the matter here is DNA itself. DNA has the ability to replicate itself. What is to replicate? To make a copy of itself. Why does the DNA have the need to copy itself? Because, why do you think? Help me out. Because the information carried in the parental cells within the DNA needs to be further conveyed to the daughter cells in an equal fashion. And we are kind of guessing and hoping that there won't be anything wrong during the process of this replication. It has a checking mechanism, obviously, and repair mechanism. If something goes wrong, goes as much as the DNA can, tries to repair things. But if in the cases where there is no means of repairing, or there is a mutation in one of the guardians of the cell cycle per se, then things start going berserk. And then you start seeing diseases such as cancer and whatnot. I will touch base on that when I start talking about cell cycle. But now let's concentrate on this. So I'm going to hold you on this little bit and then I'll move on and then start talking about in detail regarding the parts of this central dogma business. So you have here the DNA on which within the nitrogenous spaces is the information kept on. So it has the need to replicate itself to start with, but the information kept in the DNA does not mean much if you do not translate that information, right? It's like books in the library and you never go to the library to read the books. You know that they are there. You know that they have a lot of information. Nowadays, people rarely go to the libraries because they have access to the books or whatever they want to read online. So they can actually take out their cell phones and read about, from trustworthy sites, they can read about anything and everything under the sun. But there is an old school thing that sometimes people have the urge to, you know, open up a book and then turn the pages of a book, whatnot. What I'm trying to get at is that unless you go, let's say you have to go and then read something to learn something, if you don't go to the source of that information, then you will never going to get the information out of wherever it is kept. Same thing with the computers. In CPU, the information is kept, but if that information is not revealed, then it's going to be kept to itself. That's why we have go-betweens, intermediary situations, as we refer to as transcription, and this intermediary molecule is the RNA itself, or messenger RNA. And then the story doesn't end there, obviously. It has to go towards the final step, which is to get a product out of whatever is being processed. So the actual meaning that comes out of the whole story is the protein itself. In other words, the product of translation. So each and every one of these processes actually is a mean of a 1 hour lecture, but I'm trying to compile the information, make it a little bit concise, and make it a little bit easier for you to understand. At the end of the day, when I'm done with this lecturing, you're going to get the idea in terms of what I'm trying to explain you in a big picture, okay? So let's slowly move on, and then I will take part by part to explain you what I'm trying to teach you here today. So genetic information, as I have already mentioned, is stored in DNA. Where in the DNA? In the nitrogen spaces in the DNA, because it's something sacred that it needs to be kept kind of safely. They are facing towards inside. The helix, the double helix, is keeping them, so they are kind of hydrophobic. What's hydrophobic? They are scared of water. They don't like water, so they are facing towards inside. As opposed to the sugar phosphate backbone, which are polar molecules, right? The pentose sugar and then the phosphate molecule, which the DNA gets its charge from. Which part of these two molecules does the DNA get its charge? From the phosphate molecules. What is the charge? Negatively charged, right? So this entity of the DNA valency being negative comes in really handy when you're working in a wet lab situation, because it gives you the capability to be able to separate DNA fragments. You can run them on like gels. You can run them on chromatography or whatnot. So you can use the ability of the DNA or DNA fragments being negatively charged to separate the fragments. And it would give you an ease to determine what's going on. If you're working with a specific gene, you can amplify that gene. You can use PCR. If you're working with a functional assay, you can use the RNA, which is also the copy of the DNA. But you can treat your cells or animals, and then you can extract the RNA, and then you can look into what was the effect of the treatment as a result of, keeping an animal at certain regimen, or keeping a cell line at certain regimen, with treating them with different drugs and different conditions. So going back to this, there are maybe some repetitions here, but it's okay. Repetitions are good, actually. It reminds you of things that you may have missed from the previous parts, and then it enhances the knowledge, okay? So it's not because I forget what I told you and I keep repeating myself. That's not the case. The case is, and it comes from experience, the case is that I am trying to help you out in terms of remembering things, and then emphasizing on the things that I usually do these in the class, that when I emphasize on things, you understand that you're going to get it as a question in the exam. And I do say that, okay, this can come in the exam, so make a note of this, all right? So there are 20 naturally occurring amino acids, and I will show you in a table what they are. We really don't need to memorize the 20 different amino acids. If this was like a biochemistry class, maybe I would ask that you remember their structural formula as well as, their names and all that. But there are tables that you can refer to. Even there are tables for codon reading. So when you are, for example, in an exam situation, I may give you a sequence of DNA, and I may ask, let's say, transcribe this DNA piece of molecule into RNA, then translate it into protein, use the codon table, and tell me what the sequences of the like the amino acid sequence of the small protein it is, okay? And then you can do this by using the tables and whatnot, so long as you know how to use it, okay? So, again, in order to be able to produce these amino acids, again, with a mathematical formula, we can explain this. There are what we call triplets of nucleotides that represents individual codons. Codons, again, I'm jumping the gun, but it's being explained here. Codons are found in the expressing regions of the genes, which are called exons. Codons are made up of 3 nucleotides. Each codon is a specific entity for an amino acid. There may be more than one codon that can be specific for an individual amino acid. There is a unique start codon, which is AUG, which is specific towards methionine, starting amino acid, right? But it is said that genetic code is degenerate. This is an important word, the key word, degenerate. So the degeneracy of the genetic code enables less of the mutations to take place, although sometimes, you cannot avoid the mutations, but it kind of enhances or helps the system, the context, to have less of a mutation. So you may have some amino acids, and I don't have it off the top of my head right now, but you may have a single amino acid being expressed by 6 different codons. This is just to make the mutation occurrence less than, it can happen. So here, you see that, again, as I said, triplets of nucleotides give rise to a single codon. Each codon is specific for an amino acid. There are about 64 possible codons that we know of. 61 of them codes for specific amino acids. 3 of them are unique for stop codons. When the enzyme sees these stop codons, it's as if you're driving your car and you are at an intersection where there's a stop sign. What do? Usually the Turkish people, they don't exactly stop and they just slide slowly through, although there's a stop sign. But if there's a policeman there, then you get the fine. Okay, so that's the case. The next time you think twice before slowing down or stopping. And I did touch base upon the AUG here. Here is the table. It's not very readable, but what this shows you is that you have the long name abbreviations and you have the codons. And here there are the specific codons that I was telling you, that 6 of them can be uniquely be specific for the amino acids here. And then the last 3 are the stop codons. These are bases of the foundation information giving us a start to start thinking about transcription and translation. And then we're going to look into as to what we mean by this here. So the transfer of genetic information from the DNA, from the CPU of your computer, okay, into the intermediate molecule, which is the RNA, okay, is referred to as transcription. Conversion of the information, which is encoded on the nucleotides on the mRNA, into the protein is referred to as translation. If you think about what in a regular English language, what translation means, it actually makes sense, because there is a sequence which you don't know what it means. And in real life, if you are in a foreign country and you do not speak their language, and then they insist on not speaking English, like in Italy, in France, people are specific. They don't want to speak English, although they know it. There is no way you're going to understand them. Nowadays, obviously, you have these software that they can interpret it to you, but think that that did not exist. So you're just, you're going to be frozen there, no help, nothing to do. There's a language that you don't speak. You need someone. What would that someone be? A translator. Okay? So the language needs to be translated to you to make a meaning in terms of whether it be in English or in Turkish or in Farsi. Okay? It has to be converted to a kind of a means of being understandable. So that is what is being done from DNA to RNA, from RNA to protein, transcribing and then translating, okay? And information is kept in the sequences of the ACGT combination permutation, however much the length is. That needs to be transcribed first via intermediary molecule, RNA. First, the heterogeneous nuclear RNA. You remember that the heterogeneous nuclear RNA holds the introns intact. It is when the mature RNA is reached that the introns under normal, and the word normal is relative, guys, okay? Normal towards what? Compared to what? When completely in a healthy situation, when the introns are being spliced out and then the exons are joined together to form the mRNA, that is when the mRNA, the mature mRNA, is ready to go on and do its job in terms of being ready for protein synthesis. In other words, translation, okay? So this is a nice picture that I enjoy actually sharing with my students. It's an exaggerated drawing, but the exaggeration comes from being legible and then readable, okay? The nucleus should be large enough to fit the drawing so that you start with the DNA itself. Because we're talking about a eukaryotic entity here, obviously things need to take place in the nucleus, right? So transcription in eukaryotic cells, you have no other choice, but it has to take place in the nucleus, where the DNA lies. DNA, there is no way for DNA in eukaryotes to go out of the nucleus. It's always kept there, okay? That's why the mainstream thing, the transcription needs to take place in the nucleus. But what's going to happen afterwards? First, heterogeneous nuclear RNA is transcribed, then it is converted into mature RNA, and it is kind of protected by the capping and polyadenylation. Now it's ready to move out of the nucleus through the nuclear pores into the cytoplasm where translation is going to take place. But this process, this trafficking of molecules, have to be in either way. Because think about it. For transcription, you would need RNA polymerase II. It's an enzyme. Enzymes are proteins. Proteins are being synthesized where? In the cytoplasm. So how on earth is going to go back into the nucleus? Because its job is in the nucleus. It is translated in the cytoplasm, but its job, where it needs to function, is in the nucleus because it serves in the transcription mechanism. Okay? So it has to go back through, again, the way it came out of. Originally, the genome of the RNA polymerase II was, in the DNA. It was transcribed, it made into protein in the cytoplasm. So now it's ready to do its job. So it has to go back and then function as the main, let's say, main star of the process, because without RNA polymerase II, you cannot have any transcription. It has to be there. Okay? So this drawing here basically tells you how what I just said is taking place in a simple manner. We are making it very simple. It sounds very simple, but it is more sophisticated than what I'm talking about, okay? But if you get the big picture, it would be easier for you to, when you go back to a textbook or some other text, to read it. Having known the big picture, it would make your lives easier to understand what goes on in detail. Are you with me? Yes. So what components, what ingredients do you need for these processes? Well, for transcription and translation, there are need for organelles such as ribosomes. Can we say that prokaryotes also have ribosomes? Because we know that prokaryotes do not possess nucleus. They don't possess membrane-bound organelles, but ribosome is an organelle. So can we say that prokaryotes also have ribosomes? Of course. Otherwise, how are they going to protein synthesize? Right? Without ribosomes, they cannot synthesize proteins. It has to be there, okay? No other choice. So, but for the translation process, in addition to the ribosomes, which is a very important component, you also need the mRNA, because that is from which the protein is going to be synthesized. You also need transfer RNA, tRNA in short. You need the specific amino acids that are going to be bound on the specific transfer RNA, okay? You need the special enzyme which are going to couple the reactions in order to initiate these processes. And last but not least, you need the energy. Without the energy, you cannot achieve these things. So whether it be coming from ATP, CTP, GTP, TTP, UTP, whatever, the energy is there coming from the breaking down of the phosphodiester bonds out of these triphosphates, okay? That's how the energy is given out. So don't kind of narrow yourselves thinking that only the ATP molecule is the one that gives out energy. No, that's not correct. The way it was discovered that it was ATP, that chemically when they tested it, they noticed that it was an abundance of adenosine triphosphates. That's not to say that the GTP, TTP, as I said, CTP and UTP cannot give out energy. They all do. They are all capable of doing that, okay? So, where was I? Just a minute. Ribosomes, these are necessary, as we said, not only for eukaryotes, but also for prokaryotic organisms. We have the tRNA molecules that for each individual 20 different amino acid, we have a specific tRNA being involved in the action. You're not going to see me drawing the details of the ribosome that I usually give a shortcut to my students to remember. What's another word for monkey, for example? Ape. If you read ape the other way around, so A starts from the right-hand side, A, P, E. These are the regions, grooves on the ribosome onto which the tRNAs can bind and then initiate, elongate, and exit. Okay? So first, the first tRNA binds on A and it moves to the P groove. That's where the polypeptide chain is originated. And then that tRNA can move to the E ready to leave the ribosome, leaving behind the attached amino acid. And all these messages are gathered from the mRNA, obviously, because mRNA has a site to which the anticodons coming from tRNA are bound, and the specific amino acid is brought to the vicinity, and then the polypeptide is made. So think of these. I always, again, say to the students, don't just study because there's an exam involved. Try to understand the meaning, the big picture, because as I'm talking to you now and you're listening to me, you're observing me, you're looking at my movements, my hands are moving around, my mouth is moving because I'm making a sound. All these things are involved with the gene expression, okay? So we all are taking it as granted. We all are taking it as granted. It's my job to teach, it's my job to give seminars, and I like it and I enjoy it. And those of you who are also enjoying it, you come and listen from me regarding different subjects. But these liveliness, this atmosphere that we're actually putting it together, you listening to me, observing me, and trying to understand, comprehend what I'm talking about, it all involves gene expression, okay? It is when there is a problem in terms of comprehending or not being able to hear, or you're having a headache, something wrong is going on in your body, you know, because of an environmental situation, or physically you are to do something additional to fix that problem. That is when you start taking care of yourselves and visiting a doctor's office. That is when you realize that, okay, you know, I need to go and visit, and if there's a medication for this, I need to use it so that I can go back to my normal life. Okay? So then you become more appreciative, especially for those of you, most of the audience here are from medical school. So thank you guys for joining us today. We also have from dentistry students here. But the departments that you're coming from, mostly, obviously, these kind of talks are being listened by the health sciences faculties. Because you're going to be involved with humans, okay, you need to make sure that this molecular biology and genetic basic sciences are a strong foundation in your discipline. Because this is not just a course you take and then you forget about. It grows with you. Even after you become dentists, you become doctors, you become psychologists, whatever, you have to go back to the roots of understanding how your DNA functions. So that's the purpose of today's lecture, actually. It's like in a nutshell, I have to say it's in a nutshell because I haven't gone into the depth of giving you more, because each and every one of these topics involves separate lectures and separate talkings, but we have to put them under 3-hour conditions so that, you know, you get the big picture. I don't know if I can take questions, but let's move on, and maybe probably we'll have a question and answer session as well. So I already touched base about the transcription part. Somebody is no. Okay, so for the transcription to take place, you have to have an initiator, meaning a template. From something else needs to be drawn. And in this case, as you know, from the cover slide, DNA is the template. DNA is the template for RNA. RNA would be the template for the protein synthesis, okay? And there are stages where we can explain these, as the name says, You initiate the process, initiation, elongation meaning making more molecules of the thing, and then termination, when time comes for the enzyme to realize that it's time to stop because it has seen a stop codon. It means that each 3 stop codons do not have any amino acid that correspond to their, expression. So if it says a stop, it will stop. That's the end of the protein synthesis. So these 3 keywords are the stages of transcription and also for translation, for that matter. At the beginning of my talk, I was referring to the part of a gene where the RNA polymerase binds, and that part is named as the promoter, as I told you. RNA polymerase have specificity to the sites on which it can bind if it's allowed. When I say if it's allowed, if there is no hindrance, if there is no blockage for the enzyme to bind there. What would the blockage be? Methylation, for example. So CpG islands or C nucleotides can be methylated in order to initiate some sort of a gene silencing. Usually we talk about epigenetics. Epi means after the fact that you're born. Most of the time, epigenetic conditions are environmentally governed or whatever you're exposed to. So the genes can be silenced through methylation processes, okay? Here's a simple drawing just to make you understand in simple fashion how the transcription of a gene can take place. You see that the DNA is double-stranded entity. You see a big bulk molecule here, which is the RNA polymerase. To the right side, I usually don't like saying right side or left side, but I should say to the 3' end of the RNA polymerase 2, there is the actual gene. To the 5' end of where the RNA polymerase 2 is sitting is the major part of the promoter region, okay? And I didn't, here on my slides today, you're not gonna see any details regarding the components of the promoter region because the other day I was talking about TATA box, TATA nucleotides can get together to be named as a TATA box in the promoter region, which is one of the initiator sites where the first transcription factor binds. What's the name of the transcription factor? TATA binding protein. So what that does is that it goes and recognizes this TATA region, binds there, and it kind of calls RNA polymerase 2 to come and initiate something. And to that, other transcription factor joins in, and then they start the whole story. The big story starts there when the queen RNA polymerase 2 is ready to initiate its job. It sits there, everything is ready to go. There is no hindrance, and then the movement is going to start from 5' towards the 3' end of the gene. So the gene from the DNA is going to be transcribed via this queen molecule, which is the RNA polymerase 2. So, but without the double-stranded DNA opening up, there won't be any case of transcription. There needs to be an exposure of single strands for the RNA polymerase to be able to go and then, like a zipper, add the teeth. By teeth, I mean the nucleotides to the complementary sites, A to T, C to G, blah, 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 and the story goes on until it kind of looked how it is shown here. The brown color is the newly made RNA molecule, okay? So we call this a transcription bubble. There needs to be an opening up in the double-stranded helical structure of the DNA in order for the RNA polymerase to only transcribe a single strand. It doesn't transcribe both the strands. It's either the top strand or the bottom strand, okay? So you need to have this in mind. Again, as I I get excited as I teach these things over and over again, but it's like me teaching it the first time. So I live the thing when I teach these because I enjoy talking about these things. I have already touched base upon the pre-mRNA, and I told you how it can initiate in the nucleus, and then it kind of gets converted in a simple way and gets, capped and polyadenylated, ready to move out into the, again here, capping and poly-A tail. They are getting ready to move out of the nucleus through the nuclear pores into the cytoplasm. And again, I jumped the gun. I already talked about the introns and exons. Again, let me remind you, if you did not hear what I was saying, introns are the parts of the genes which are not involved in the expression mechanism. They get spliced out, but the lecture that I will be giving you, actually the last lecture, I will be talking about mutations that give rise to the disease itself by the splice mutations, not having a complete splicing of the introns, but some parts of the uncoding regions are left behind in the mature mRNA, and that can be deleterious in terms of the outcome of the disease. So when you compare genotype with the phenotype, you can actually look into the types of mutations. When I'm going to be talking about the beta thalassemia, from an experienced eye, we can actually talk about the specific mutations either being a severe condition as a phenotype or less severe or intermediary condition. And then the MDs, the hematologists, can decide whether the patient in question would be put into the regimen of receiving blood from the donors, okay? So it has to go hand in hand with the MDs and the molecular biologists, because it used to be that the university that I graduated from was the first reference laboratory, Boğaziçi University in Turkey, in the whole Turkey, that we did the first molecular analysis of certain single gene disorders, such as beta thalassemia, Duchenne muscular disorders, hemophilia, cystic fibrosis. So there were different groups involved in these single gene disorders, and we were the first ones to initiate prenatal diagnosis, which I will mention when I'm going to be talking about the beta thalassemia, okay? So when I teach these topics, I would like to give a relevance in terms of why would we care what happens, at the DNA or RNA or protein level? It has to have a connection to the health or disease so that you can compare. Why does that disease take place, and in what condition it takes place? What are the means of correcting that disease? And I will give you examples of that when I talk about beta thalassemia. As opposed to the introns, the exons are the parts of the mRNA which actually are involved with the codons, which are the coding regions, which have the amino acids that can be involved in the polynucleotides or the proteins themselves. Here's a nice drawing actually showing you the initiator, which is the DNA. This is kind of inverted DNA from 3 to 5. We usually draw them from 5 to 3. And then the transcription gives rise to the first part of the RNA molecule, which is the pre-mRNA. This is when the capping and the poly-A tail addition takes place. And then finally, these sniped out parts are the introns. The light brown parts are the introns. When they completely get cut out, then they enable the exons to join to each other to kind of form the mature mRNA, okay? And then the mRNA is ready to leave again the nucleus, going to the cytoplasm to initiate its function in terms of protein synthesis. Now, it used to be in the old books that I was taught actually, that one gene, one enzyme hypothesis. This is no longer true. What do I mean by one gene, one enzyme? What they meant was there's a gene for one enzyme, but we now know, thanks to having established the sequences through Human Genome Project, and we have like 30,000 some genes expressing, it doesn't mean that we have only 30,000 proteins being produced from these genes. Actually, the number is 100 or 200 or a little bit more, thanks to the alternative splicing. What does the word alternative means? Other choices, right? You have other choices to show yourself. So these genes can be alternatively spliced to give healthy outcomes by, let's say the original gene has 12 exons in its pre-mRNA, and it can give rise to different kind of a muscle, either striated muscle or a muscle that would be a smooth muscle. So different type, it's still going to be a muscle, but different types of muscles. So the variety is going to change thanks to the alternative splicing, meaning that there may be more than one type of protein that would be specific to the single gene because of the alternative splicing, okay? I will touch base on this in more detail, but very quickly going through it. We will talk about the splicing defects in human genetic disorders. As an example, the beta thalassemia is a hemoglobin disorder. And this is actually one of the drawings that I use when I talk about beta thalassemia. The beta globin gene itself is located on chromosome number 11. It's a family of genes. So all the genes are like groups of genes that are similar in terms of having same number of introns, same number of exons, but the sequences may be different. The only thing is that there are two different types of gammas which only change A gamma, G gamma. Alanine and glycine is the, or glutamic acid, I think, is the amino acid which is different from one another. Otherwise, they are the same. There's a pseudo beta, there's a delta, and all these names of these boxes, the colored boxes that you're looking at, they each represent a gene within the gene family of the beta globin gene, okay? And these express themselves, again, I'm jumping the gun and telling you before I start talking about the thalassemias, but these genes expressed in correlation with the alpha components, the embryonic, the fetal, and then the adult versions of the proteins. We will talk about them when time comes, but this slide was just to give you an idea as to when you have mutations, these splicing mechanisms may change, and as a result, you get mutated protein, and as a result, you get a disease, a genetic disorder called beta thalassemia or alpha thalassemia, but we will concentrate on beta thalassemia today. So, as was the case for the transcription in terms of translation, mRNA, amino acids, ribosomes, tRNA, and energy sources are needed in order to be able to produce 20 different amino acids. Okay? Again, the list. So let's just change the gear a bit and then talk about the structure of the amino acids, just to have more meaning, just to go a little bit in more depth rather than just the name. So we hydrogens, and then carboxyl group is COOH. If you were to be asked to give a structural drawing of the amino acids, this is how you would do it. There's a carbon atom in the middle. You know that the carbon can make 4 bonding. You have a region, what we call a side chain, depicted by R. You have a part where only hydrogen binds. And then on the left and right, you have the amino group, NH2. And then you have the carboxyl group, COOH. So you can represent the O part with a double bond, and then OH here, okay? So this, you can start naming the amino acids by changing the R group. 20 different changes will give you 20 different choices of the R group. Are you guys cold? Because I am cold. Can we turn this off? Just a minute, sorry. Şeyde mi gitti? Geldi mi? Sorry guys, we're freezing cold in here, so I had to turn off the AC. Okay, let's move on. So the 20 different changes on the R part of the amino acid structure is going to give you 20 different choices of amino acids. Okay, And then these amino acids, due to the R groups, can either be charged in terms of negative, positive, or neutral, okay? So can you guys hear me? Gitti mi niye? Bununla ne alakası var bağlantı? Gidiyor. They can hear me. I think, I hope. What does this have to do with the teams? Huh? Yeah. I'm not sure whether you can hear me or not. You can hear me. Okay, thank you. I'll go on. Okay, so thank you, I got it. So I would like now to introduce you to the big picture of how is it that two different or two of the same amino acids can bind with each other to form polypeptides. Poly means many different peptides. So the linkage between carboxyl and amino ends of each of these can enable the binding and joining and forming of the peptide bonds. So we touch base upon the N-terminus, the NH2, C-terminus, COOH. This is how it is done. So you have two of the same or two of the separate amino acids, of which the amino and carboxyl ends can react and then give out, as you can see, what do they give out? Water molecule. And as a result, they make a bond between the two amino acids, which is called a peptide bond. This can go on depending on how many codons there are, because for each codon there will be a specific amino acid, and then it would be determined by the message, the coding of the message. When the enzyme reads till the stop sign, then it will determine whether, the stop codon doesn't have an amino acid, so the process is going to end there. So the whole thing is going to be kind of fulfilling the meaning of translation, giving certain length of a polypeptide formation through the translation process. So this is one of the nice questions that I always like asking in the exam. So, okay, we said that not only eukaryotes, but also prokaryotes also need to have ribosomes, because without ribosomes, you cannot achieve translation, period. Okay? So there are, I didn't go into detail here, but there are two components of the ribosome, small subunit and large subunit. I'm not going to go into detail, but the movements of these subunits can enable the movement along the mRNA as to go up until the stop codon and then finish up the translation process. So codons are placed on the mRNA, anticodons are placed on the tRNA. They have to match in order to be able to do like a zipper. It has to move on. So, remember the word APE. So joining on the A first and on the P, the bond is made, then the tRNA moves on to E, and E is for exit and just, gets out of the, the ribosome, and then the next one comes in. Then the story goes on up until the stop codon is reached. So that would be the end of the polypeptide chain synthesis. Just to show you, an upside down tRNA is joining with AUG, the start codon, which is methionine. So UAC would be the anticodon part. Okay, so the methionine part is the amino acid is going to be carried at the 3' OH end of the tRNA. So this OH is not just for decoration, it binds the amino acids, okay? So 3' OH end of the tRNA is the part where the amino acids are bound. Again, there is a nice picture of this, but I don't have enough time, so I didn't show you a lot of things. So I'm trying to wrap up the big picture, okay? If you're interested, come and visit me and I can talk about it to you. I have no problem in explaining you in detail. Okay, so again, as I was saying, initiation, elongation, termination, there are parts where the mRNA, the initiation of tRNA comes in, small ribosome subunit moves along the mRNA, and then the first step in translation begins. The start codon, as I was saying, is AUG, specific to methionine. The story goes on up until the enzyme reaches the stop codons, UAG, UGA, and UAA, and which means that it has to stop. There is no other choice, okay? Unless there's a mutation there, and it doesn't know how to stop. It's like you see the stop sign, you come to stop, but you cannot stop completely. Maybe there's something wrong with your brake of the car. You push it, but it doesn't stop, so it moves slowly, and you just go through, and you get caught by the police if there's a police around you, and you get your fine, but you can explain yourselves that there was something wrong with your car, but there is no choice of the enzyme to explain itself if it cannot stop the process of, adding another amino acid. So that would give rise to an anomaly in terms of the health of the individual, whichever gene it is kind of in question, okay? So I had to mention this because there are different levels of proteins when they are being expressed. That's not the end of the story, okay? So the proteins, in order to be functional, they need to be able to fold into the correct way that it needs to fold to be functional, okay? So there are primary, secondary, tertiary, quaternary levels of protein folding. So one needs to know these, or the proteins themselves, the big picture needs to know this to be able to function accordingly so that there is no abnormality towards the end. I'm going to end here and then start my next slide, which is about the cell cycle. Let's see. Let me try to load this. So I chose these topics as to, they're actually linked to one another. We started talking about the nucleotides, then I started talking about the packaging that has to do with the nucleotides. Then I started talking about this regulation of gene expression. You need to know this. When we say in general, like the central dogma, it involves not only the transcription, but also the translation. Although I talked about things in a nutshell, it gave you the idea in terms of what is involved, okay? Because every one of these topics involve like a lecture of their own, so we don't have time for that. Let's see. This is also another interesting topic that I like talking about with my students, because this actually opens another door to genetic disorders such as cancer, although I will touch base on back and forth when I'm talking about this, but I usually link this to a topic of cancer so that I give examples in terms of different cancer types. So the cell cycle. When we talk about the cell cycle, what do we exactly mean? What does it do when it cycles? What is the need for the cell to cycle? Mitosis, right? Wound healing, repair, growth. These are some of the components that requires the cell cycle to function in its entirety. So we're going to have a look at them, and I will give you some tips as to what goes on where and how. So I'm just making an estimate or guesstimate in terms of the timing of the whole cell cycle. Let's say it takes 24 hours, okay? The 20 hours of the whole process would be spent in interphase, okay? Only the 4 hours, like 1/5 of it, is going to take place in mitosis. And just to remind you, old school book information, interphase is not part of mitosis. Interphase is the time spent longest in the cell cycle. During the interphase, as it was the old school information, the cell doesn't rest. Actually, it's the busiest time, okay? You don't see visibly under light microscope things going on, but if there was a means of observing them chemically or whatnot, you would see things rushing around, okay? Trafficking in and out, blah, blah, blah. So molecules moving around because they are getting rushing, they are getting ready like little ants trying to find food and then take it into their, hives or their homes. The chemicals go around looking for the correct component to do this, to do that, to complete the process of preparing, for example, what's going on? Huh? Okay. So the cell itself is preparing for division, but in order to make things okay, or work for the goodness of the cell, number one, you're talking about a parent cell, just one parent cell. What does that need to do? It needs to prepare itself to divide into two to start with, but it needs to do it properly to divide equally so that one doesn't get more than the other one. So if you have 46 chromosomes in a single somatic cell, it has to make double the amount of that. It has to increase its volume in terms of the periphery of the cell membrane. Otherwise, how is it going to fit all those doubled amount of organelles, the DNA, and whatever is needed to, pass it to the next generation? Everything is doubled in terms of content, and the size, the volume gets larger. Because when it divides, it has to be equally the same, okay? So this is what the process is doing during the interphase. But anything and everything may go wrong while these things are being done. When I say may go wrong, things can get out of hand. So all the steps needs to be under control. So what would control them? Do they have a policeman? Do they have any guardians? Do they have something that actually controls this? Yes. There happens to be what we call, and I'm jumping the gun again, happens to be checkpoints. Like as if you're traveling, and those of you who are in Cyprus, the traveling is, especially with the plane, you have to go to the Ercan airport, okay? So I'm making an analogy now. What would you need to fly from here to Istanbul, for example, or from Cyprus to Tehran, to Iran? You need something to show when you're leaving the country. That something is a valid passport, a valid ticket. Even if you have one of them, okay, you won't be able to fly because, number one, if you have a valid ticket that you have an expired passport, they are not going to let you go through. So somebody at the checkpoint is looking at these things. In other words, there is someone which is checking for the correctness of you being able to fly as a legal passenger to your destination. It's similar to what goes on in the cell. At the checkpoints, and before I come to the checkpoints, let me tell you the phases first. So there is a G1, S, G2, and M phases in its entirety regarding the cell cycle. G for gap, okay? S for synthesis, M for mitosis, okay? So you see this gray area here, it takes the longest time, and it is the interphase, the preparation stage for the division, before the division of the cell. So in order for me to show you, okay, this next slide is actually going to tell you where the checkpoints lie and how the things are controlled under the mechanism of cell cycle. So let me give you the names of the checkpoints as well, and then go back and tell you shortly what happens in a single phase, and then we will take it from there. So the first checkpoint happens to be not exactly at the border of the S, but a little bit further down to the left into the G1, and it is called G1/S checkpoint. So that's the checkpoint, like as if you're in the airport. before you fly. That is the checkpoint that is going to check whether everything is, going well for you to proceed further. So if everything is well, and I will tell you what those everything are in a minute, you can pass, not you, but the cell can pass into the S phase. S for synthesis. What is it going to synthesize there? DNA. In other words, DNA needs to replicate there so that the same amount of nucleotides or whatever is coded on the DNA can be passed on to the next generation, okay? So then comes the second checkpoint, G1S, G2M. That's exactly where the border is. You can see here before it moves on to the M. And there, again, some questions are being asked to the processes. Are these being kept okay? Or then you're ready to move on. And then the last checkpoint comes within the mitotic phase. It says M, but M is not for mitosis. M is for metaphase. Why? Because during the metaphase part of the division of the cells, the chromosomes lie at the equator or at the center of the division part where the spindle fibers are joined to the centromeres. So that is being checked because there can be defects like trisomy 21, okay? Although that happens during the meiosis prophase 1, but I'm just trying to give you an example. If the spindle fiber doesn't bind properly and when they're pulling towards the opposite poles, the chromosomes, one may stay behind. So either of the daughter cells is going to receive either one less or one more number of chromosomes, which is going to give rise to some abnormality as a result, which would give rise to some sort of a genetic disease, right? So this is what's being explained. So going back very quickly, so we did mention about the checkpoints here, 3 different checkpoints, G1S, G2N, and M. So in the G1 phase, the questions chemically asked are, the cell growth, has the cell grown to adequate volume so that it would keep the increased number of organelles? Duplication of the organelles. This is another question that is being asked. Preparation for DNA replication. So DNA replication doesn't occur as quick as, when the DNA decides to replicate. There needs to be preparation for the presence of the content of the replication, such as the primers, the enzymes, the proteins that are involved. Okay, DNA needs to be unwound and this and that. So a lot of things are involved before the DNA starts to replicate. So have all those things been fulfilled before it passed to the S phase? So these are things that are getting ready in G1, and the questions are asked at G1S checkpoint, whether all these things that are indicated here are kept or fulfilled, and the process continues through the S. Again, there, the DNA is synthesized, and then another question in G2 is being asked at the checkpoint, again, if everything is okay to pursue along. So these simple questions make a lot of sense when it comes to defining and finding the errors that have been made in the previous phase, so that the enzymes or the proteins have a chance to go back and then correct them. It is when the guardians, and I will name them in a minute, and I will give you the specific names of these guardians. It is when they cannot do their job, and then the abnormalities initiate to pursue. So there are 2 major events during the cell cycle, and these would be your S phase and M phase. This just summarizes the processes during mitosis. So before mitosis is the interphase, as we said. Then there's the prophase, metaphase, anaphase, and there are in between phases as well. Telophase and cytokinesis. Karyokinesis first, and then cytokinesis, the division of the cell in itself. So some scientists like to study certain phases using FACS analysis, meaning that they use this technologies where they can accumulate the specific cells at specific phases, such as this one here, which made a peak in the G1 phase. They can collect all the cells in G1 phase. Then, what I'm talking to you about, what goes on in G1, what goes on in S and G2, that information is obtained from this kind of research, okay? So they can obtain G1 phase cells, S phase cells, G2 phase cells, and whatnot, and then they can study this particularly and ask whatever question they want to ask. So I did touch base about the contents, but this is important that one needs to know regarding the cell cycle. There are two major components. What makes the guardians be able to work are, these are cyclins, cell cycle dependent proteins, and cyclin dependent kinases. As the name says, kinase, okay? These are enzymes. What do kinases do? Their job is to add phosphates. They phosphorylate. Kinase enzymes help in phosphorylation, but they don't work on their own. They have to work together with cyclins, and cyclins work together with the kinases. So cyclin-dependent kinases, the name as it says. In this drawing graph, you see the different phases of the cell cycle, you know, G1S, G2M, and then it just shows you which kind of CDKs and cyclins are involved at each process. You can see these increases, sigmoidal appearances, plateauing, and then going down the threshold, meaning that the production, the duration of whatever they're doing, and then decreasing in terms of the concentration of the particular cyclin-dependent kinases. Here, now I want to talk about these, again, major components of controllers. In other words, the checkpoint police. P53 is the major one, tumor suppressor gene. The name says tumor suppressor because in its entirety, positive meaning, it suppresses the tumors. But when it cannot do its job, and in most of the cancers, they have detected the mutations in the P53, meaning that it will be a mutant tumor suppressor gene. So it won't be able to do its job. So we need to add the word mutant so that we know what we are referring to. Because in the context of your conversation, if you are all scientists, then you may understand each other, whether you're talking about the positive sense or the negative sense of the tumor suppressor genes. So whenever we talk about the people who work with cancer, talk about tumor suppressor genes, we usually refer to the mutant version of it. But when you talk to students or people who doesn't have much idea about these things, you have to say mutant tumor suppressor gene because it doesn't do its job because of the mutations that take place in its genetic structure. So what you see here are, these are also tumor suppressor genes, P4 protein, P16, P27, P21. They are the helpers of the queen tumor suppressor gene, which is P53. Together with that, they have a control on the CDK4 and 6. So these are two separate kinases, CDK4, CDK6. Together, they join with the cyclin D1 protein. So together, three of them match, and then they can control at the checkpoint whatever goes wrong, okay? So if there is something wrong with these CDK4, 6, and cyclins, then these tumor suppressor, these inverted T's show that they inhibit the function of that so that the cell cycle doesn't go on. Because if something is wrong, the proteins at the checkpoint needs to have a control over the cell dividing so they can stop things, they can look into how they can correct it, and if there's a means of correcting whatever went wrong, then they can correct it, and then, you know, the phase movement clockwise, it just goes on. There is another important tumor suppressor gene called retinoblastoma, in short RB, okay? It causes tumors in the eye, especially in the kids. It doesn't happen in the adults because it happens in the embryonic cells. So once this retinoblastoma protein is phosphorylated by this CDK4, 6, and cyclin D1, then it can release itself from what he binds onto E2F protein, and that E2F protein has a function of initiating gene expression of certain genes down the line. So if there is no fault in terms of continuing the cell cycle, then the process is such that RB will be phosphorylated by CDK4/6 and cyclin D1, and then it will release itself from what you see on the table here from E2F, and then E2F is going to continue as a transcription factor to bind on the promoter region of whatever gene that needs to be bind on, and then the cell division is going to continue. But this can also happen in the case where there may be mutations in one of these genes that it cannot do its job, or RB itself can be mutated. It is a tumor suppressor gene, but it can become mutant tumor suppressor gene, so it won't be able to bind onto E2F, so the cell cycle is going to unstoppably continue, which is going to give rise to what? Tumor formation. Uncontrolled cell division is the name of the game here. Cancer is initiated by the uncontrolled cell division because of these wronggoings in the cell cycle phenomena, okay? So, in a nutshell, it goes on like this. Again, you're all welcome to, ask me questions through email, or you can come to my office, those of you who are in EMU, and ask me these questions. But you see how fascinating it is? You never think about these things, right? You only come to the realization when something is wrong. But it's nice to think about the, normal version of life in terms of everything going well, and then it makes more sense. So, how are we doing with time? More to give like a break? Tamam, okay. So I'll try to be short and almost finished actually. Let me see. Yeah. So now I'm going to give you some examples regarding the cyclins and CDKs. What can they do and what names they take and how they get phosphorylated or dephosphorylated. So on the left-hand side of this drawing, you see a cyclin-dependent kinase, which is colored in red. On top of that, there's a phosphate molecule, and this phosphate molecule is an activating phosphate, okay? And then the green part represents a cyclin, any cyclin. I'm not giving it a number, I'm not giving it a name. So this is like a generalized version of the process. So going from left to right, the V1 kinase, and I told you at the beginning, kinases are the enzymes that phosphorylate molecules, okay? Sometimes phosphorylation can be advantageous or disadvantageous, or let me put it this way. Phosphorylation can make something active or inactive. The same thing as dephosphorylation. Let's have a look what it's going to do in this case. Well, you can see that at the bottom, an active molecule is being made inactive by the V1 kinase phosphorylation of the molecule on the right-hand side. So this phosphorylation is an inhibitory phosphorylation. So it inhibits the process, so it inactivates it. But the other way around, so in Chemistry 101, you know that the reactions, some reactions can go either way. So this is one of those kind of reactions. If you go from inactive towards the active, what would you need? You can use a CDC25 phosphatase. What do phosphatases do? They remove phosphates, unlike the kinases, okay? So by taking this molecule that I'm pointing at with my cursor and then moving towards right, the other way around, you're going to see that CDC25 phosphatase removes one of the phosphates which initiated an inhibitory process. And then the molecule is going to be active. Are you with me? I mean, if you go slowly and then do it like a scenario and think about like a big picture, what it would do, it would all make sense. This one here, we're using the tumor suppressor P21 as the example. The molecule on the left-hand side is an active molecule, cyclin and CDK complex here. There's already a phosphate molecule, but it doesn't matter. So going from left to right, once the P27 senses that something goes wrong, it goes and binds onto that molecule, that CDK and cyclin component, and then it inhibits it itself, because its role is that, its function is to inhibit things that are going to cause a wrong processing down the line. So once the P27 tumor suppressor binds onto this molecule, it inactivates it. So in the process, maybe it should inactivate it, because down the line, if it doesn't get inactivated, it's going to cause for the cell cycle to continue progressively, non-stop. And we don't want that, okay? It should be under control. This list here is like yeast experiments. In order to be able to find a function of a molecule or a gene or a protein, in molecular biology and genetics, what we do is that we create transgenic animals or knockout animals. So what we do is that we actually create mutations in certain genes, and then we observe the offspring, whether it would be viable or it would have some sort of abnormality, or we kind of study its function depending on what goes wrong, okay, or what is beneficial. And this table, I'm not going to go through the whole thing. Once created, these animals can either have sterility, they don't have babies, or they die in uterus, or any abnormalities due to the fact that we create these mutations in their systems. I touched base on P53, but you should know that this is a protein that senses DNA damage, and it halts, it stops the progression when there's a damage in the DNA for the cell cycle. But in order for this to be mutated and non-functional, both the copies, one coming from your mother, the other one coming from your father, needs to be mutated, okay? So mutation means it can have a point mutation, substitution mutation, frame shift mutation, or deletion. Any one of these on one gene should be occurring, and on the other allele also should be occurring. So you cannot have one normal allele and the other one mutated allele. That would be heterozygote, and it would not function in terms of being a tumor suppressor, in terms of mutant being a mutant tumor suppressor gene. Okay, so in order for this gene to be held accountable for wrongdoing, both of the alleles, both of the copies should be mutated so that it doesn't function in a healthy way. You should have asked me, I talked about G1S, G2M, but I didn't say anything about G0. So there are times, and it's not very important for the cell, but there are times that scientists want to be, we all are curious, and we want to study what happens if the cell is in its dormancy, like hibernating animals, not doing anything, non-functional, or quiescent form, that, we study cells when they are not doing anything. What's going on there? What is the important thing that we need to know that the cell is kept in G0 state? So that's for the curiosity's sake. And thank you.